Swayam Prabha. Digital India. Educated India.
We all had some fun thinking about the stable marriage problem and got some taste of game theory. Okay, so now what we'll try and do is try to see how some of these ideas applying to understanding biology and in particular understanding ecosystems. I, in particular, do research on ecosystems and in particular understanding how we can think about ecosystems as mathematical objects, as using mathematical principles, okay? But before I tell you about ecosystems, I want to tell you a little bit about this. Has everyone heard of this? Okay. Who all is here familiar with tide? Okay, good. Everyone knows. Excellent. Okay, what is tide? It's a detergent. Okay, so I want to tell you a little bit of a story about tide. Okay, about the detergent Tide, which is that they actually got into a bunch of trouble in the 50s and 60s with governments in North America and in particular Canada. And the reason for that were these things. So many of you have heard of Tide, but who here has heard of a cyanobacterial bloom? Oh wow, a non-negligible number of people. Okay, so for those of you who have heard about it, what is a cyanobacterial bloom? Yeah, it's an algal bloom. What is an algal bloom? Okay, good. So, and people have seen algal blooms in Chennai and other places around you. Okay, so what do you think is the link between cyanobacterial blooms and tide? Okay, yeah. So this was part of a major legal battle in the 50s, where at that time it was still not clear what causes algal blooms. I would say some people will still say it's not clear what causes algal blooms, but one of the big culprits at the time that the Canadian government thought was detergents. Okay. And in particular, the thing they thought was bad about detergents that caused these cyanobacterial blooms. So this, by the way, just to give you a sense, is this lake called the Lake Erie. It's one of the big lakes in North America. It's ginormous. It's so ginormous that do you know what these things are for scale? These are like, yeah, this is like towns and buildings, right? And so what you can see is that all of this green over here is big growth of alga. And this is a problem. Does everyone know why algal blooms are a problem? Yeah, so first of all, it kills everything in the lake beneath it because it sucks up all of the nutrients and the oxygen. Blocks sunlight. Is it bad for us? So actually, many of these algae release insane toxins into the air, which many of, like, especially in North America, if you're going around these algal blooms, you can get massively sick if you don't have like a proper mask around some of the algal blooms, not all of the algal blooms. Yeah? So this was becoming a public health hazard. People, of course, could not take dips in these lakes, and just generally a lot of places in the US and Canada rely on these lakes for water. So that was another huge problem. So suffice to say, this was a big issue. These algal blooms were not just an aesthetic problem. Okay, and the problem that the Canadian government at the time thought was with tide was what this one nutrient called phosphorus. Have people heard of phosphorus? Okay, and it turned out at that time, Tide and other detergents of its type, Tide was just the biggest one, happened to have a lot of phosphorus in it. Okay, can anyone guess why there was a lot of phosphorus in the Tide? So I didn't know about this, but apparently it was to soften the water. Yeah. So they put a ton of phosphorus in it, and what Tide's contention was, and what a lot of other detergent companies said, was that it was not the phosphorus that was causing the problem, and so Tide was not at fault for causing these algal blooms. It was something else. In fact, they said it was something like nitrogen that was a problem. So at that time, the Canadian government employed this guy, Stephen Carpenter, and gave him basically access to these extremely pristine, gorgeous lakes. They look like this. They had access to a few hundred tiny ones of these types. Some of them were quite big. This is now called the Experimental Lakes area. And they gave him this big kind of free hand to say, you can do all the ecological research that you want with these lakes. You can even manipulate some of them if you want. But we want a clear answer. We want definitive scientific evidence showing that it's phosphorus or it's nitrogen that's causing these algal blooms. Okay? So the task is clear, the task that's set up for David Schindler and Stephen Carpenter. So what do you think they did? They had access to many of these lakes. What do you think they did to actually figure out whether it's nitrogen or phosphorus? Maybe we can raise our hands and go one at a time, just so that it'll become easier for me to hear. Yeah. Okay, so you put nitrogen, and then let's say if it grows, then where's the phosphorus? Yeah. Excellent. Very good. Perfect. So remember I told you there were like hundreds of these lakes that were available. So they had access to one such pair, and basically the idea was that you take one of these lakes, to one of them you add nitrogen, and to one of them you add phosphorus. You see what happens. Okay? And so here's a photograph that is actually very famous now. This was taken from a helicopter. These are two different lakes. This is actually a road or a street that's separating these two lakes. That's how close they are. They're basically the same lake, and they were in fact very similar and clear before this experiment happened. But now you see they're not, right? One of them is still clear. One of them is turbid, right? It's full of this bloom. Okay? Can you predict which one had which? Which one did he add? Which one did they add phosphorus to? Which one did they add nitrogen to? Okay, wait. Let's take a vote. So for those of you who think they added nitrogen to this lake on the right, raise your hand. Okay. So if they add nitrogen to this lake, they add phosphorus to this lake. Yeah. So one is NP, the other is PN. So we're doing the NP first. So which of you think that it's N on the right and P on the left? Okay, and which of you think it's P on the right and N on the left? Okay, the majority of you. So most of you think it's nitrogen that's causing the turbidity. Yeah, you're with Tide. Okay, so what do you think they actually found? So they found this. Okay, so the turbid lake, they had added P, meaning this lake was clear before, they added P, it became turbid. The clear lake, the other clear lake, they added N and nothing happened, right? So what do you think this established? Phosphorus was a big component that was causing these algal blooms. Why do you think, by the way, why should any of these nutrients, including phosphorus, be responsible for these algal blooms? Provides nutrients to the alga, right? But nitrogen is also a nutrient. Okay, that's a good guess. They might need phosphorus more than nitrogen. So by the way, do we know what all of us are made of elementally? Carbon, that's it. Nitrogen, phosphorus, hydrogen, oxygen, trace amounts of magnesium, calcium, any other big, tiny amounts of all of these things. So CHNOP S, sulfur, right? These are the 6 big ones. Okay, let's just talk about carbon, nitrogen. So which of all of these are we the most made up of? Carbon, that everyone agrees, right? What is next? Nitrogen, phosphorus, oxygen, what? Okay, so how many of you think oxygen? Not so much oxygen. Okay, maybe let's talk about nitrogen and phosphorus. Do you think we have more nitrogen or phosphorus? So actually we have 10 times more nitrogen than we have phosphorus. Okay, and we have 10 times more 10 times more carbon than we have nitrogen. Okay, so roughly each of us are like 100 parts carbon, 10 parts nitrogen, 1 part phosphorus. Okay, and so are many of the organisms in these lakes. Okay, so they actually need nitrogen more. They get it from the atmosphere. It's a good thought, but actually most of the atmospheric nitrogen is very, very, very hard to access. It's true, most of the atmosphere is nitrogen, but most of it is very, very hard to access. So actually it turns out that the nutrient that you are made less of, that you have less of, is actually what's limiting your growth, right? So if there's a lot of nitrogen and very little phosphorus, if you raise the amount of phosphorus, you're going to be able to grow a lot more. If you have a lot of nitrogen, it doesn't make too much of a difference to your growth, right? So both of these, nitrogen and phosphorus, are nutrients that organisms need to grow, but phosphorus is usually what is limiting your growth because it's available in smaller amounts in your environments. And so the idea was, if you provide more of this, you tend to just suddenly explode in your growth. And that's exactly what was happening to these lakes. Okay, and just to cut that story and bring it to a close, that this is ultimately what was the clincher that gave the Canadian government the free hand to then regulate Tide and other detergent manufacturers to reduce the amount of phosphorus that they had in them. And indeed, the Tide that you and I use today indeed has a lot less phosphorus than it used to, thanks to this experiment. Okay, but this experiment was amazing at the time, not just because it showed this thing and had this practical application, but because it also showed something very, very crucial about the ecosystems around us, which was that, these ecosystems can usually be very, very stable. So, these lakes can stay clear for a long, long time. But every now and then, if you perturb them, if you give them a disturbance, and that disturbance you can give by adding nutrients, you can shift them to another state. And this other state also turns out to be very, very stable. What do I mean by stable when I say stable? If you try to move it around a little bit, it doesn't change. It's not very easy to change. Okay, so lakes, once they become turbid with these algal blooms, they actually stay turbid for a long, 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 long time. Okay, and so this is what kind of gave us the idea that in nature, the ecosystems that we see around us, they can be in different stable states. Some turbid, some clear. Some other cases, there could be some other things. And there are perturbations that are very targeted. In this case, adding phosphorus but not adding nitrogen, that can trigger transitions between states. Yeah, this is a thing that we agree that we learn from doing this. Okay. We have since then been able to take this idea not just all the way from lakes, which are big, big objects, but also to small systems. So these are, does anyone know what these are? Terrarium. It's a good thought. These are not quite terrariums. What are terrariums? They have plants and stuff, right? Yeah. Has anyone heard of the word Vinogradsky column? Okay, when I was in school, we used to learn about Vinogradsky columns. But okay, so this is possibly the simplest science experiment that you can do. And you can do it today if you want. You take one of these jars or a measuring cylinder or a cup or anything that can be closed, okay? You go and you scoop out some soil, okay? You mix it with some water with a spoon. Maybe if you want, you crack an egg into it to provide some more nutrients. And then you close it. Okay, the closure is very important. So these also, you see, these are all closed. And you just let them sit on the window counter so that they feel the light, the day and night cycles. Okay, if you just leave it for a couple of months, this is what you'll see. Sometimes, isn't this beautiful? So what do you think all of these different colors are? You'll see different colors every time you do a different experiment. These are different kinds of bacteria and they're producing different little pigments that are causing this beautiful coloration. And all this is saying, like, what does it mean now that you see different sets of colors in different columns? They're different sets of bacteria, right? They're different states or different stable states that you find in each of these different columns, okay? Here's another example. Do you know anywhere? So these are all also ecosystems because there are many different species of different bacteria. Do you know anywhere else other than the soil where you have systems of where you have ecosystems of microbes? In your own body. Where in your own body? In your gut, but also everywhere, including everywhere on your skin, in your mouth, everywhere. But those of you who heard of gut microbiome? Okay, what is the gut microbiome? Sure, yeah. So basically the idea is that we eat food. A lot of the food is really complex and humans do not have the ability to digest it. Can someone give me an example of one such food? Yeah, what is cellulose? Like, just give me an example of some food you actually eat, right? Yeah, any plants, most plant matter, yeah, almost all plant matter, we don't have the ability to digest on our own, okay? We don't possess the enzymes to do that, okay? But many of us eat plant matter all the time, right? So how is it that we are able to actually get any nutrition from this plant matter? We don't have the enzymes to digest this plant matter, but there are tons and tons of bacteria in our guts that do. Okay? And it's actually only because they can break up this complex plant matter for us that we can actually digest any food, especially vegetarian food. Yeah? This is very important. Okay? For us. We also know that, for instance, these kinds of microbiomes or microbial communities can be very diverse. Okay, they have very many different kinds of species in them. Can anyone guess, for instance, in this little scoop of soil or in a little scoop of your gut, how many different bacterial cells there would be? Who thinks it will be less than 100? Okay, less than 1,000, less than 10,000, 100,000. Okay, some people think it's 100,000, a million. Okay, some people think it's a million, a billion. Okay, more than a billion, tens of billions. Yeah, so it's actually trillions of different bacteria in your gut. What is a trillion, by the way? It's 10 to the what? 10 to the 12, right? Billion is 10 to the 9, million is 10 to the 6, okay? And so what you'll see typically is, well, that's in our gut, but just a single gram of soil can contain up to a billion different cells of microbes, okay? And of course, like, you have many grams, you have tens to hundreds of grams. And each of these different cells are very many different species. So they're very diverse, thousands to tens of thousands of species of different microbes. This is not just true for the soil, it's also true for our guts, it's also true for the oceans, it'll also be true for that door handle over there. They're everywhere, okay? That doesn't mean that they're bad. Like we just discussed that the whole reason we can digest most of our food is because of them, right? And so, this is what we were just saying, that our gut microbiome is also an ecosystem of thousands of species. It controls our digestion, and it does so only because there are these thousands of different species that are interacting with each other and doing all sorts of stuff, okay? And just like we were talking about in the last lecture, these gut microbial communities or gut microbiomes are also in some stable states, and they can also transition between these multiple stable states. So here is an example of some data from an experiment. Okay, so how many of you have seen data plotted in this way before? Okay, good. So we'll all learn something new today. So on the x-axis here, I have time in days. Okay, so what we are doing is we are tracking someone whose gut microbiome we are able to access. How are we able to access people's gut microbiomes? We sample. How do we sample? Yeah, so when you poop, 50% of poop is the gut bacteria that you have, right? So it's a very easy way to collect these gut microbial samples. So we're tracking someone over many, many days. You can see that actually we already started tracking them a long time ago. So this is already starting at day 120, 130, 140, all the way till 190. So, you know, for almost half a year, if not more, right? And what we are plotting on the y-axis here are how many, like how much each of the different species of microbes are in it, okay? So each of the different colors here, the green, the yellow, the orange, and so on, are different species of bacteria, okay? And just for simplicity, I've shown you only 5 of them, okay? So what do you see here? That over time, these species of bacteria kind of stable, right? Their abundance or their population size is not changing with time too much, right? Everything is stable. We'll call this state 1. Now, this person actually happened to get an infection, okay, in their tummy at around day 150. Okay, and I marked that over there. What has this infection done to their gut microbiome? It's changed the which bacteria are there at what population sizes, right? So what do you see? Can you tell, by the way, which of the species did the best as a result of the infection? The orange species did really well, right? Very infectious species. And which species had a really hard time? Yeah, the blue or the purple species had a really hard time. It basically went extinct, right? And now we'll, you know, this infection lasted for 10 to 15 days. Maybe some of you have experience with this. What do you think happened after the infection cleared? Do you go back to state 1? Yeah. So in this case, you went to a new state, to a different state. We'll just call it state 2. You can see that these 2 states are different. The orange species is very high abundance. The blue species is kind of gone. You can see there are also some species like the green that was at low abundance, came up and came back down again, right? Just something that happened transiently, right? There are also some other species like the yellow that again changed a lot during the infection but came back to where it was before. But suffice to say, at the level of the whole ecosystem, this state is different from this state. Do we all agree? Yeah. Not all the species are completely different from what they were like before. State as a whole is different. Okay? And so this is a phenomenon that we see all of the time, not just in gut microbiomes, not just in the lakes, but in many, many other situations where you make a transient perturbation to an ecosystem and it completely changes in what state it has. Okay? And so the question that we were asking here in this work was, how can we understand or how can we model this fact that there can be multiple stable states? So is it clear what I mean by multiple stable states? Okay. And our answer in this case, which I'll tell you about now, was competition for nutrients. But before I tell you the answer, can you make a little bit of a link between the first lecture and this lecture? What were we talking about in the first lecture? Stable marriage, and in particular, what aspect of the stable marriages were we talking about? Compatibility, stability, no incentive to change. And in particular, in the end, what did we kind of stop at computing? We computed some stable states and we learned that there could be multiple stable states, right? You see the same idea we're trying to explain here. How can there be multiple stable states? In this case, we are not caring about men and women. We are caring about microbes or different species. So I'm going to try and show you that these two problems are actually related or connected with each other. Okay? So here is how they can be connected. So what you often see in these gut microbiomes is that different species often compete for the same nutrients. Okay, and I've tried to represent that here in the form of a network. How many of you have seen a network before? Okay, never seen a network before. Great, so we'll also get to see what networks are like. So in this case, I've connected all these little different wiggles of different colors are different species of microbes. This is what we were tracking in the person who just got infected. And on the top, I've unimaginatively named all of the different nutrients. Okay, just to give you a sense, this C is what some of you were calling cellulose, right? This H is actually something which is a smaller form of cellulose called hemicellulose, and so on and so forth. Okay, names are again not so important. Whoever told you in biology the names are very important, they're not. I don't even know what these colors at the bottom mean. Okay, but the important thing to note are these connections. Yeah, so everyone sees these connections. What do you think the connections represent? Yeah, it represents which nutrients these different species can eat. So for instance, this purple species can eat essentially all of the nutrients, right? Because there's a link or a connection between the purple species and every other nutrient. Can you spot a species that doesn't eat too many nutrients? Yeah, this green one only eats 2, right? M and P. The yellow eats 4, right? And this cyan one here only eats 3, right? But typically what you can see is that there's a lot of competition. How do I see that competition? There are many nutrients which many different species can eat, right? And so they must be competing in the gut for all of these nutrients. By the way, all of these nutrients are names of different things that we eat, right? Like cellulose, hemicellulose, pectin. P is actually turns out to be something called pectin. Again, the names are not so important, but I'm just trying to point out these are like real examples of real things happening in your gut right now. Okay? So there's a lot of competition for nutrients, right? And at the same time, do you think that these species, so you see that here species can eat many different nutrients. Do you think if the purple species was presented with all of the nutrients all at the same time, like a buffet, would it eat all of them at the same time? No, right? Probably when you also get access to a buffet, you eat things one at a time, right? So this is exactly what microbes also do. That they typically use nutrients one at a time. Okay, and usually that means they have a preference hierarchy built inside them, right? So here's an example, again a very classic example from a bacterium called E. coli. Anyone heard of E. coli? Very famous bacterium, right? So here what we are tracking in red is the population size of the E. coli bacterium over time. So the y-axis is the population size measured in some way, and the x-axis is time. Okay, you can see 0 hours, 3 hours, 6 hours, 9 hours. And you can see in red, what do you see? This E. coli grows, right? Its population size increases with time, but it grows in two phases, right? There's phase 1 and phase 2 written there. What's different about the two phases? Correct. So initially, we presented this E. coli with two options. We gave it both glucose and lactose, two different nutrients, okay? And it grew, right? But in this blue, we're also tracking how much glucose there is and how much lactose there is. And what do we see? Initially, when the E. coli grows, only the glucose amount goes down, right? The lactose amount stays the same. After some time, the glucose has run out, right? What does the E. coli do now? Now it starts eating the lactose, right? And in fact, do you see that there's a little bit of a delay in between when it finishes using the glucose and it starts to use the lactose? Why do you think there's a delay? Yeah, exactly. So it's trying to transit, like it's the E. coli is trying to transition from eating one thing to the next, right? And that requires it to change inside its cell all the different things it needs to do to be able to eat this lactose, right? So that little delay over here presents that, right? So what does this E. coli prefer more and what does it prefer less? It prefers glucose to lactose, right? So we now know that actually if you did this experiment with a different species, not with E. coli, but with some other species. Does anyone know the name of any other species of bacterium? Lactobacillus, sure. Streptococcus, sure. If you did this with any of these other species, what do you think you will observe? Will they all prefer glucose to lactose? No, actually some of them prefer lactose to glucose. And actually you can do this not just with two nutrients, but with many nutrients. You'll find all sorts of different hierarchies of preferences for which species prefers which nutrient more and which nutrient less. Okay? This is also true of the microbes in your gut, these microbes that we were considering before, right? So by now we know that the microbes in our gut and everywhere, they're all competing for many nutrients, and when they get access to multiple nutrients, they often have a preference hierarchy. They often have a preference for those nutrients, right? And actually this results in a very simple model and a very simple way to understand how to make sense of all of this stuff, which is that you have two different actually ranked tables which encode the preferences of each of the bugs of each of the species, and that encode how competitive they are when they are trying to compete for each of the nutrients. Okay, so here's an example of what that could look like. So say I have this red microbe, which I'll call M1. I can have a situation where it prefers the nutrient N1 the most, N2 the second, and N3 the least. It can happen, right? For the same nutrients N1, N2, and N3, the yellow bug might instead prefer N3 the most, N1 the second, and N2 the least. It can happen, just an example, right? So I can encode all of this in a very simple table where the rows are the different species and the columns are the ranks, are the ranked ordered things of how competitive they are, right? Okay. At the same time, I can also look at the competitive abilities of each of the microbes. When the yellow and the red microbes competes for nutrient N1, it could be that the yellow bug wins. When they compete for N2, maybe the red bug wins. And when they compete for N3, maybe the yellow bug wins again. And I can also encode this in a simple ranked table where now the rows are the different nutrients And the columns are again the ranks of which is the most competitive and which is the second most competitive. And I've ended up with 2 ranked tables or 2 ranked ordered preference lists. Do you see what is coming next? Does this look like the stable marriage problem? I don't have men and women. What do I have instead? I have microbes and nutrients, right? I had preferences for men, which were different women. I have preferences for, do I have preferences for microbes that are different nutrients? Yes. We also had, remember, preferences for women, which were different men. How do we have preferences for nutrients here? That when 2 microbes go and propose to a nutrient, when they go and try to compete for this nutrient, one of them will be better and will win. One of them will be worse and will lose, right? So does everyone agree that this is kind of looking a lot like the stable marriage problem, right? And so indeed, we took inspiration from the stable marriage problem to try and map this. So we did all of this before. We had these lists. We talked about stability. We talked about the notion of blocking or eloping pairs, right? And we also talked about this men proposing algorithm, right? So what we didn't just run for the Pride and Prejudice case, we can run for now, which is, we can run the women proposing algorithm here. So on the first day, all of the women go for Bingley, one of them goes for Wickham, so two of the women get rejected, they go down, there's more rejections, you go down until everyone's matched. Okay, we already did this, so we can skip over this, but everyone's clear. You can go ahead and apply this algorithm to find at least one stable pairing. So this is exactly the analogy that I was bringing up. What used to be a set of men and women are actually now microbes and nutrients. And what used to be women's preferences can be thought of as maybe nutrient preferences or competitive abilities of microbes. And what used to be men's preferences can thought to be the preferences of different microbes for different nutrients. Okay? What does this allow us to do? What could we do with the stable marriage problem that is actually going to be useful here? What's our goal? Is to understand whether there can be multiple stable states and finding those stable states, right? That's exactly what the stable marriage problem gave us. It gave us an algorithm to find not just one stable state, but many stable states, right? So now we can play a few different games and try to understand what this model is trying to tell us about microbial communities, okay? So one of the things that it tells us is how you can change communities or make them transition from one state to another by making a transient perturbation. Remember when we were talking about lakes, we were talking about a perturbation with phosphorus taking you from one stable state to another. Okay, so now again we'll try to ask what can we do in this case. So say that we start off with this situation where these are my preference lists, the same ones that I constructed before, and I have 2 nutrients, nutrient N1 and N2. I don't have nutrient N3 present right now, and I have a stable state. I have the yellow species eating nutrient N1, and I have a red species using nutrient N2. Can confirm that this is a stable state. What I'm going to do is I'm going to transiently introduce a new nutrient, okay, this N3. What is that going to do? Once a new nutrient is available, all of the microbes will look for whether they prefer this nutrient to the one they're currently using. Yeah. In this case, will the yellow bug actually prefer nutrient N3 to N1? Yes, you can see in this list that the yellow bug prefers N3 to N1. Okay, so it will try to move to eating nutrient N3. And now at the end of this, N1 is free. right? No one is eating N1. What does a red bug do? Does a red bug prefer nutrient N1 or nutrient N2? N1. So it'll try to move over to eat nutrient N1. Okay? And now, if at this point of time I take away nutrient N3, it was only transiently available, then my yellow bug will, what will it do? It'll go to N2. Why will it go to N2, not go back to N1? Because it prefers N2 more, right? So N2 is free, it will go to N2. And at this point, even if N3 is not available, we have entered a new stable state. This state is also stable. Both of the bugs get their top choice, right? So what did we do? We started off with one stable state, one in which both the bugs did not have their top choice, right? But it was still stable. And we ended up by this transient perturbation of introducing nutrient N3, ended up with a new stable state where actually both of the microbes have their top choice. Okay? These kinds of restructurings could happen in our guts. Here's another example. You can play the same game again, where you now don't introduce a transient nutrient, but you introduce a transient species, another species, in this case a purple species, that will come in, it will try to invade. We will see that it will be unsuccessful in invading, but even though it will eventually be knocked out, it will have changed the stable states. Okay, so here, let's say we start off with the case where both of the bugs are the best. The red bug is eating N1, the yellow bug is eating N2, they're both eating their top choice. I introduce a new species, this purple species. What does it prefer the most? It prefers N2, and so it will try to eat N2. Is it more competitive on N2 than the yellow bug? Yes, so it will knock out the yellow bug. The yellow bug will go to nutrient N1. Will it knock out the red bug? Yes, it's more competitive on N1 than the red bug, right? So now the red bug will go to nutrient N2. Will it be able to knock out the purple species? Yes. And now the purple species is gone. Okay, at this point, if the invasion is unsuccessful, you have now switched over. Now the yellow species is using nutrient N1, and the red species is using nutrient N2. This is also a stable state. In fact, it's the same stable state that we had at the beginning of the last slide, right? So again, you can have a situation where you can have a transient invader come in. It'll eventually go out and lose, but it can still cause a shift in the stable state. So if you're saying at this point, if the yellow goes to N2, it will not be able to. That's why it's a stable state. That's exactly what stability is. In this case, so here it's simple, right? Because both of them have their top choice. They have no incentive to switch, right? Here they have incentive to switch, right? The yellow bug wants to switch to N2, but it cannot. It's less competitive. So it's also stable, right? You have to find a microbe and a nutrient who would want to be with each other more than they are with their current partner. Okay? Yes?
Why can the purple microbe go back to N1?
The purple microbe, we could imagine, could go back to N1. In this case, what would happen? It prefers N2 second. Is it more competitive than the yellow bug on N1? No, so it would lose, right? So this purple microbe is just stuck now, yeah? And that is in this case because you had only 2 nutrients, right? But 3 different microbes, right?
What if it's stuck in like an endless loop and stays?
It could be, but usually you also poop thankfully. So everything is fine. Eventually, if you're not growing by eating nutrients, you're going to get washed out. Yeah. So in fact, this is, how many of you have heard of probiotics? Have any of you taken probiotics? What are probiotics? Curd is a probiotic, but what is probiotic about curd? They actually have bacteria inside them, okay, but why is that a good thing? Yeah, the claim is that those bacteria actually help you do a lot of things in your gut. But actually there's a lot of times in which you eat curd every now and then. You eat a little bit of the bacteria. If those bacteria don't manage to grow well in the gut microbiome, if they happen to be like this purple species that you introduce into your gut, but that is actually unsuccessful in invasion, it can still have an impact, right? It can still change the state of your gut microbiome, even though the bacteria inside that curd might not necessarily stick around for very long. Yeah? And this is also one of the ways in which now people think that probiotics work, right? So have probiotics. Okay, so I will cut a long story short. You can go and take now more complicated cases where, in this case, there are 7 microbes and 7 nutrients. And you can use these algorithms that we talked about in the first lecture, the men proposing algorithm, the women proposing algorithm. We'll just call them now the microbe proposing algorithm and the nutrient proposing algorithm. You can do the break marriage situation where you kind of transiently change and orphan one of the species in what nutrient it's using. And you can essentially find all the different stable states. And just to kind of point it out to you, a stable state here is just an enumeration of which species is eating which nutrient. Okay? So in this case, for this example that I constructed, I won't go through it. You can show that there are only 5 stable states. Out of how many? 7 factorial, right? That's a huge number of states, but a very, very tiny fraction of them are stable. Yeah? And again, you can kind of show how you can go and transition from one stable state to another. So this is just a more complicated example of now 7 species and 7 nutrients, where you can transiently remove a nutrient, just like we were doing a few slides ago, and cause the stable states to shift. Okay. And so in this way, you can not just predict all of the stable states. You can also predict how can you transition from one state to the next. Remember when we were talking about lakes, we talked about examples of 2 different perturbations. What perturbations were they? Yeah, it was either adding P or adding N, right? And only one of the perturbations was successful in transitioning the stable state. Which of them were they? Only the phosphorus one, right? So in this case also, what you can show is that only some of the perturbations, so only some of the times you add a nutrient, will you actually be successful in changing the stable state. Okay, so you can make another network. This is an example of this network in yellow, where each of the different nodes of the network are different states. Each state was one of these columns, which species is eating which nutrient. So I can call states S1, S2, S3, S4, and S5. So I have all of my 5 states over on here. And I have now arrows that direct me from one state to the next. What do you think these arrows mean? Yeah, these arrows are telling you how you can make perturbations to transition between states. So the way to read this network is, in this particular case, you can go from state 1 to state 3 by doing what? By transiently removing nutrient 7. Okay? But if you transiently remove nutrient N5 instead, you will go to state 2. If you transiently remove neutron N3 instead, then you go back to the same state, right? So you can see that most of the states, most of the perturbations that you make, 5 out of the 7 neutrino perturbations you make to state 1, the state is going to be resilient. Nothing is going to happen. Only 2 targeted perturbations are going to take you from one state to another state. Same thing here from S3. The only perturbation you can make is by transiently removing N5. You can go to state 4. From S2, you can only transiently remove N1 to go to state 4. And then finally from S4, you can only transiently remove N4 to go to state 5. Any other perturbation is going to keep you where you are. Okay? So by perturbing so this way not only gives you all of the different stable states, but also tells you which kinds of perturbations you can make if you want to go from one state to another. Then this could be important, right? You can imagine that there could be cases where you want certain species to be in high abundance versus low abundance because it's healthier for you. And so this also tells us how to do that. Okay, I will end here and sort of summarize. I hope you saw and I hope you can see that understanding properties of ecosystems is both fun and challenging. We need all sorts of new approaches to think about how to model them, and it's a lot of fun. We can use a lot of mathematical tools, computer science tools, physics tools to understand and study these kinds of systems. Not just ecosystems, I would say, many different biological systems without ever having to remember any names of chemicals and molecules, knowing what the species are, etc. And then finally, we can use ideas from game theory to understand which ecosystem states are stable and which ones are not. Okay? All right. So I will stop here, but if any of you have any questions, I'm happy to take them. Thank you.
Running a little bit behind schedule, so maybe we'll do the quick tea break now. If any of you have any questions for Akshit, you can approach him during the tea break. We'll be happy to address them. He'll be on campus today till around
3.